The second stage of photosynthesis is light-independent reaction, which is also known as Calvin cycle or C3 pathway. C3 is named after the first stable product can be formed in the Calvin cycle, which is the 3-phosphoglycerate, which is the 3-carbon compound. And the Calvin cycle or C3 pathway is very common in C3 plants, such as legumes, potatoes, wheat, and paddy. The light-independent reaction occurs in the stroma of chloroplast, which is the fluid space found in chloroplast. And it happens during daytime or daylight, but the light energy or photon is not required throughout the Calvin cycle process. It happens during the daytime because they require the ATP and NADPH molecule that produce from light-dependent reaction. And light-dependent reaction occurs during daytime because they require energy. The importance of Calvin cycle is because they are going to reduce the inorganic molecules, which is carbon dioxide, to become the organic complex molecule, which is the glyceraldehyde 3-phosphate, a 3-carbon compound. They are going to be combined and produce a 6-carbon sugar, which is the glucose. The light-independent reaction Calvin cycle is divided into 3 phases, which begins with phase 1, carbon fixation. We have briefly studied these terms in the carbon cycle of chapter ecology. The second phase is reduction because it involves the NADPH, which is the reducing agent produced from light-dependent reaction. The last phase is regeneration of carbon dioxide acceptor, which is the ribulose bisphosphate. Theoretically, we are going to study how one molecule of glyceraldehyde 3-phosphate, which is the 3-carbon organic molecule produced from the Calvin cycle. Since this molecule is 3-carbon, so when we study about the carbon fixation, we always begin with 3 molecules of CO2. Let's begin with phase 1, carbon fixation. And the molecule CO2 is an inorganic molecule that contains only 1 carbon atom. Why we start with 3 molecules of CO2? Because as I say, at the end of carbon fixation, the molecule CO2 will be reduced into the organic molecule, which is the glyceraldehyde 3-phosphate, a 3-carbon compound. So to produce glyceraldehyde 3-phosphate, we need to fix 3 molecules of CO2. But each Calvin cycle can only fix 1 CO2. So to fix 3 molecules of CO2, the Calvin cycle have to happen 3 times. And how does CO2 can be fixed? It's actually accepted by carbon acceptor, which is the ribulose bisphosphate, or abbreviation RuBP. Each RuBP is a 5-carbon compound with 2 phosphate groups. The fixation is catalyzed by the enzyme RuBP carboxylase or Rubisco, and it produces 3 molecules of 6-carbon intermediate, which is unstable. Each 6-carbon intermediate or the 6-carbon compound that has 1 carbon origin from the CO2. With the presence of 2 phosphate groups in 1 6-carbon compound, they make this molecule reactive or unstable. So immediately it will split into 3 carbon molecules, which are 3-phosphoglycerate. It means that every 3 molecules of CO2 fixation is able to produce 6 molecules of 3-phosphoglycerate, which is the stable molecule. And this is the reason why Calvin cycle has another name, which is C3 pathway, because the first stable molecule is 3-carbon 3-phosphoglycerate. The second phase of Calvin cycle is reduction, but it begins with the step that each 3-phosphoglycerate will be added with one inorganic phosphate group from the ATP. ATP are the products produced from light-dependent reaction, includes the non-cyclic and cyclic photophosphorylation. With the addition of one inorganic phosphate group, now the molecules becomes 1,3-bis-phosphoglycerate. The next step of reduction is reduction step, which means each 1,3-bis-phosphoglycerate is reduced by reducing agent in the photosynthesis, which is NADPH. Is another product produced from non-cyclic photophosphorylation. So, NADPH will donate the hydrogen ions and electrons to the substrate 1,3-bis-phosphoglycerate and become NADP+. With the reduction to this molecule, it becomes high energy. So immediately, each of it will remove one inorganic phosphate group. This is why we have 6 inorganic phosphate group released from the group of 1,3-bis-phosphoglycerate. And they will become a reduced compound, which are glyceraldehyde 3-phosphate or G3P. Each of these 3 carbon molecules carry one inorganic phosphate group. So during the phase 2, the reduction phase, we notice that every 3 molecules of CO2 fixation able to produce 6 molecules of G3P. But only one G3P will be used as the starting molecule to produce the glucose, which is the 6-carbon organic compound. And they will undergo other metabolisms process to produce fatty acid, glycerol, sucrose, or other polysaccharides such as starch and cellulose. The one molecule of G3P used as the starting molecule, it can happen randomly. It doesn't mean that must be the G3P carrying the C from the CO2. And the remaining of the 5 molecules of glyceraldehyde 3-phosphate will move on or proceed to the next phase of Calvin cycle. In the last phase of Calvin cycle is the regeneration of carbon acceptor, which is the RuBP. So we start with the remaining of 5 molecules of G3P from second phase. They are going to undergo rearrangement of atoms through a complex reaction, which involve a few steps and the step catalyzed by enzymes. And at the end of phase 3, it produced 3 molecules of RuBP. Each of it is 5 carbon compound, and they are going to be used in the next Calvin cycle. During the regeneration of RuBP, we notice that 3 ATP molecules are used, and at the same time, they are the removal of 2 inorganic phosphate group to produce the 3 molecules of RuBP. The reason why 3 ATP molecule is used to form 3 molecules of adenosine diphosphate and with the removal of 2 inorganic phosphate group is because if we notice the population of the 5 molecules of G3P, they consist of 15 carbon atoms here and 5 inorganic phosphate group from this population. But at the end of the phase 3, there are 3 molecules of RuBP to be formed. Each of it is 5 carbon compound. So in this population, they have 15 carbon atoms as well. But if you notice carefully, each RuBP carried 2 phosphate group. That's why they have the name bis-phosphate. So we have 6 inorganic phosphate group here. Okay. So from 5 inorganic phosphate group, to form 6 inorganic phosphate group, they need to be catalyzed by a series of complex reactions. With the addition of 3 ATP, that means 3 inorganic phosphate group has been added to this population of intermediate molecules. 5 inorganic molecule plus 3 inorganic phosphate group, we have 8 inorganic phosphate group. But to become 6, they have to remove another 2. This is why the addition of inorganic phosphate group comes together with the removal of 2 inorganic phosphate group. So to produce 1 G3P, which is always the starting molecule to be used by the plant cell for other metabolism, the number of Kelvin cycle need to occur 3 times, because each Kelvin cycle can fix only 1 CO2. So to produce 1 G3P with the 3 times of Kelvin cycle, they require 3 molecules of CO2 and also 9 molecules of ATP, which required in the phase 2 and the phase 3, and 6 molecules of the reducing agent, which are NADPH, that required in phase 2 also. But in plant cell, glucose is still the most important sugar to be used for other metabolism as well. So to produce 1 glucose with 6 carbon, we need to produce 2 molecules of glyceraldehyde 3-phosphate, because 3 carbon plus 3 carbon is going to produce a 6 carbon molecules. But the details on how the 2 inorganic phosphate group can be removed from this combination is not in our syllabus. Okay. So if we study how many molecules of CO2, ATP, and also the NADPH to be required to produce 1 glucose, we just need to double the data from the production of 1 G3P. So as a conclusion, photosynthesis consists of 2 stages which occurs separately, but they depend on each other. This is because the Kelvin cycle, they require the ATP molecules and the reducing agent, which are NADPH, that can be produced from the light-dependent reaction. And at the end of the Kelvin cycle, they are going to form ATP and also the NADP+, which is now the oxidizing agent. These 2 are required in the light-dependent reaction. Both stages happens in chloroplast. Just the difference is Kelvin cycle occurs in the stroma of chloroplast, and light-dependent reaction occurs in the thylakoid membrane of the chloroplast. When 2 molecules of G3P are produced from the 6 times of Kelvin cycle, they are going to be combined to produce 1 glucose, which is the 6 carbon sugar. And the glucose can be converted to form fructose, which is also the 6 carbon sugar. They can combine with the glucose molecule to produce sucrose, which is the transported sugar in plants.